Assalamu alaikum wa rahmatullahi wa barakatuh. Peace, mercy, and blessings of God be upon you. Welcome to a new emergency presentation from www.central-mosque.com and from the Humble Heart YouTube channel. And the topic today is Mufti Amjad, killing babies, and coronavirus vaccine. There was a program in Dewsbury on the 20th of March 2021, and Mufti Amjad Sahab Hafidahullah Ta'ala is being accused that Mufti Amjad knows that vaccine has aborted cells, abortion is murder, so vaccine is totally haram. I have not been asked by Mufti Amjad Sahab to respond on his behalf. He has not deputed me to respond. But I know the anti-vaccine brigade and the fanatics, and to be honest, they take pride in their ignorance. I am from the US South, and I know the Christian right-wing nut jobs when it comes to abortion is murder tagline, and it is designed to be emotive. I have spent much of my life around such people, and some of the YouTube watching British Muslims are simply regurgitating the ignorance of right wing Christian nut jobs, parrot fashion. So let me try to answer the ignorance from this channel about the objections on Mufti Amjad Sahab Hafidahullah Ta'ala without his knowledge, permission, input, or consent. The issue is about the Islamic ruling regarding the Oxford AstraZeneca COVID-19 vaccine, which is detailed in Fatwa 029 at the Wifaqul Ulama website. Link is in the description. If they would have just read the fatwa properly, perhaps the whole controversy would have been avoided and all of our times would have been better spent. Let me read the text from the document under the heading Aborted Fetal Cells. MRC5 are a group of cells that can be grown in a lab or outside of the tissue and originally taken from an aborted fetus in 1966. AstraZeneca disclosed to full fact that the available vaccine does not use MRC5 cells. The vaccine, however, does use HEK293 Trex producer cell line. Let me repeat, cell line, which were originally derived from kidney cells from a female fetus aborted in the 1970s. None of the originally fetus-derived cells are used in the vaccine, and purification was performed by cesium chloride gradient ultracentrifugation to remove the cell culture material. The fatwa clearly states that the vaccine does not contain fetal cells. The cells which are used are cultured in the lab and grown from the original 1970 sample. Let me repeat, the vaccine has no fetal cells or baby cells. That is the first point. The second point, it very clearly states that it is not impure. Now let me address their point of not being impure even though the cells are lab grown. So cesium chloride gradient ultracentrifugation was pioneered by Matthew Maselson and Franklin Stahl around the 1950s. I think it was 1958 or something. They invented this to separate DNA labeled with different isotopes of nitrogen. These different isotopes of nitrogen have slight differences in density. In this picture, you can see nitrogen isotopes of different densities, and you can see N14 and N15, which are isotopes of nitrogen. You can see different densities right here. When you place cesium chloride in water, the ions dissociate. So looking at this picture, you have the cesium ion, which is positive, and the chlorine ion or chloride, which is negative. So this molecule of cesium chloride right here, when it's put into water, substantive amounts of water, it breaks down and breaks. The ions break again into a positive and a negative ion. Why does that happen? Because if you remember from your school chemistry days, the water molecule also is negatively charged and positively charged. And there are loads and loads of this water. So what it does, it gets in the middle of the cesium chloride and breaks it apart. So then what happens is the cesium is positive, so it gets attracted to the oxygen part of the water molecule, which kind of looks, it makes a little bond with oxygen. By the way, the same thing is happening to the chloride, which is negative. So it is also being attracted to the water molecule but it's getting attracted to the hydrogen and not the oxygen. So in simple terms, the cesium chloride is being torn apart by water. Why? because the simplest way I can explain is that cesium chloride, or CsCl, is attracted to each other with some force, but there's so many water molecules pulling at it that it overcomes the force in the middle of this cesium chloride. The force that the water is pulling at it is greater, so it rips it apart. So now, if you put cesium chloride in water and you simply leave it there, so for example, if you take a glass of water, dissolve some cesium chloride, it will be ripped, but it will be evenly distributed from the top of the glass or whatever container you're using, top of the glass, middle of the glass, bottom of the glass. It will be split apart and it will stay there. However, what Maselson and Stahl did was they put it in a centrifuge. You must have seen in the hospitals and labs, when they take your blood and they put it in a centrifuge. This is a machine that looks like that. And what they do is they spin it at a very high speed. You must have seen this machine, at the doctor or the hospital. So this is what it's supposed to do. It's supposed to spin, 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 spin. Okay. In this particular case, when they're talking about the vaccine in the Wifaqul Ulama fatwa, it's very clearly written that they're using gradient ultracentrifugation, which means that they're using insane speeds of spinning. So if you take a look at what happens when you spin this for days, I'm talking about days at extreme high speed. So before centrifugation, imagine if it looks like that. After centrifugation, it just gets separated into layers like this because it is being spun at very high speeds. Let me now get back to cesium chloride and what is happening. So what is happening is the cesium chloride is broken. It's ripped apart by the water. However, the chloride is a pretty light. It's about, the weight is about 35. So even though it's spinning at really, really high speeds and gravity is working on it, it's pushing it apart, but it doesn't really have a massive impact because it's not that heavy. So what happened is the chloride ion stays pretty much everywhere in the tube. So it gets evenly distributed from the top of the tube, bottom of the tube, middle of the tube, and it's fine. But what happens is the cesium ion, the positive, right here, is quite heavy and it's around 133. So as it's spinning, so imagine if you're on, if you go to to a fair or something and you're spinning around, you will get sucked to the center of it. So what actually happens is the cesium starts to sink. So as you get to the bottom, because it's spinning at really high speeds, gravity is sucking on it, and remember, it's happening for days, sometimes for a few days, 2 days, 3 days, or even 10 days sometimes, right? And it's acting at 100,000 times gravity. Okay, and it's sucking this cesium to the bottom, sucking to the bottom of the tube. So more cesium goes to the bottom, keep sucking, keep sucking. So there's some still at the top, but most of it gets down to the bottom. Let me show you. So this picture shows you the stuff at the bottom is a lot denser, right? So that is your cesium, you can see in this image. This is called a concentrated gradient. So let me explain. More cesium down at the bottom. Why? Because it's heavy. As the tube is spinning and spinning and spinning, it's being sucked at the bottom. So there'll be more cesium at the bottom, less in the middle, and some at the top. So this is called a concentration gradient. What in the world is a concentration gradient? Let me give you a very simple example. Have some tea, put some sugar in it, and the sugar sinks to the bottom because it's heavier. So as you keep drinking your tea, imagine your cup of tea, sugar falls at the bottom. So the bottom of the tea is more concentrated than the top, and then that's why your tea at the bottom generally is sweeter than the tea at the top. So what's actually happening is a machine, something like this, is spinning that for days at extreme high speeds. So what happens to cesium? So we started out with the cesium chloride solution right here. After spinning for days and days and days, what it becomes, it completely rips apart the molecule that you started off with, and you can clearly see protein at the top, DNA at the bottom, DNA, sorry, somewhere here, and RNA at the bottom. It is completely ripped it and separated it. So if you then take out the DNA or the RNA, it does not contain the protein. So let me give you a very simple example. If you had some kind of najasa on your hands, and what you do is you go wash your hands with soap and water, and the Sharia now considers your hand to be clean. This is happening at an industrial scale in this machine. The DNA or the RNA of the vaccine is separated using cesium chloride gradient ultracentrifugation, which is a process of spinning the tube for days and days at super speeds. You have forces that are 100 times, I'm talking about 100,000 times gravity hitting a test tube. So this is what happens, and this is the reason why, even though the fatwa very clearly states that the cells are not from a fetus, they're lab grown, but even those lab grown ones, the najasa of them has been ripped apart and separated. So this is what it looks like, which was never a baby cell or fetal cell to begin with. Jazakallahu khairan. Assalamu alaikum wa rahmatullahi wa barakatuh.